Hi, and welcome back. Upscaling biomanufacturing in gene and cell therapies is one of the biggest challenges of our industry. An efficient supply chain will help us bring these therapies to patients. I'm happy to invite Dr. Gregory Thiel, Director of Biomedical Manufacturing Network, to lead this discussion. Gregory, please take it away.
Thank you, Amir, and I'm very happy to be here, and I'm joined with an outstanding panel today. I represent the Biomedical Manufacturing Network, and I also represent NIMBLE, which is the National Institute for Innovation for Manufacturing Biopharmaceuticals. And I'm joined today by Chuck Calderaro from KITE, Alison Moore from Allogene, Joe Gold from the City of Hope, and Bob Dean from Synthego. And so I have four outstanding people to share their insights. And so I'm going to jump right in, and we're just going to start with the question of what's going on right now in this space as far as advancements, but also challenges. There's been lots of talk about cell and gene therapies. There's been lots of interest from investors, from entrepreneurs, from companies that have been interested in being part of this part of the biopharma industry. So what are some of the challenges we're facing? And we'll start first with Joe. And Joe, maybe you can give us some insights, especially because you work with small companies. After that, Alison will give us some insights from her company, then followed by Chuck and then Bob. And so each of you can give us some insights about just what's going on right now in cell and gene therapies, but more importantly, what are some of the challenges that you think we're facing? So go ahead, Joe.
Thank you, Greg. So from my standpoint, because we're taking on early stage projects where we're getting things into phase 1 trials for the most part, doing some process development. The challenges that people tend to run into immediately, first of all, have to do with scheduling. There's a big wait list to get vectors produced pretty much anywhere. Certainly my facility, it's probably 12 to 15 months right now. Cell therapy projects also, there's a wait list, so people have to take that into consideration. Other challenges are that people really need to think about the process they want to do and materials they want to use. And the best thing they can do to kind of help with that is to come to a place like City of Hope or other places early, a GMP facility early, and get some input as to the methods you want to employ and materials, because we can give you some tips on alternate sources or things you may want to avoid or things you may want to attempt just to smooth out the process later on. So, my role really is to try to prevent landmines from being planted in a process which will go off later. And so I want to work with you early to avoid these kind of things. And those include the materials, the processes, and just the scheduling, realistic scheduling, taking into account how much time it takes to get something done, but more importantly, how long it takes to get something fully release tested.
Great. Thank you, Joe. And as Joe pointed out, the City of Hope offers resources to help companies, to help people interested in cell and gene therapies. And so part of what we're doing here with LA Best is introducing us as individuals. So if you want to reach out to us, so any of you in the audience who want to reach out to any of us to get help, to get insight, to just talk about the topics we are experts on, then that's one of the benefits of LA Best. In addition, if you have any questions or you'd like to ask, please feel free in the question and answer part of Zoom, and then we'll try to get those later on in the hour. So Alison, if you could take it from here and talk a little bit about the journey for Allogene and just what you see as some of the challenges and maybe some of the ways you have overcome them or are overcoming them.
Thanks, Greg. Yeah, I would call out two things at this part in our conversation. I think the greatest challenge across the whole cell and gene therapy space remains our inability to very well describe critical quality attributes and our methods of analytical understanding. So I think that we're fortunately a growing sector and we're making lots and lots of progress there, but that remains something that's quite challenging across the whole industry. Another big challenge in the cell therapy space to this point in time has been scaling up appropriately. So I will absolutely let Chuck speak to that in the autologous space. But probably our audience understands that in the autologous space, the options are really scale out options. At Allogene, we're using allogeneic biology to allow us to use source material from healthy donors, and that allows us to make 100 or more doses from each donor. So that actually has been very interesting because it allows us to marry some of this new cell therapy process biology with a little bit of traditional biologics process biology. So we actually do have some mechanisms of scale up available to us. So, but those are the couple of things that I would throw out at the moment.
That's excellent. Thank you, Alison. That really helps frame it as far as just better understanding as well as how to scale out. We'll look to both Chuck and Bob to talk a little bit more about scaling out. And then you can chime in later. We'll have another chance for you to talk a bit more about that too. Chuck, if you could take it from there and pick up a little bit on some of the challenges, but also if you see some additional challenges in the cell and gene therapy space, that would be very helpful.
Oh, thanks for the question, Greg. And it is a very challenging and exciting time in cell and gene. We have over 100 startup companies, 300 clinical trials in all various stages. So it's a really exciting time for the technology, and it's great news for patients. Regarding the scalability challenges with autologous CAR-T, I think Alison hit it. I mean, we have to bring the product and the manufacturing to the patients. It's the epitome of personalized medicine. We start with the patient's cells and need to return those cells that we engineered back to the patient. That's a 17-day for us in the United States, end-to-end turnaround time. So as we scale, the challenge is really on our logistics and distribution, connecting for us over 220 apheresis centers to 2 commercial manufacturing facilities and 1 clinical facility. So being able to do that consistently, reliably, and efficiently every single time is something that we take a lot of pride in, and we work very hard to ensure what we say chain of custody and chain of identity to make sure that the patient gets back their cells and not somebody else's, and their cells don't go where they don't belong. So our main manufacturing facility in Southern California supports all markets, and then our relatively new facility in Amsterdam supports Europe, Middle East, and then we have our 3rd facility, which will be licensed middle of next year on the East Coast of the U.S. to kind of pick up additional volume and growth, and not only in the U.S. but also in South America and such. So for us, the challenge is continuing to build out the apheresis treatment centers, that network, and then supply chain distribution logistics to move the cells and the product back and forth. And these are exciting times.
Great. Thank you, Chuck. That really helps frame it out for the audience. And those of you who are just joining, both Chuck and Alison have really described what we're facing very graphically for cell and gene therapies. And the way it's different than traditional biopharma processing, where we have a few locations that we scale up and make lots and lots of doses of something. Instead, we have truly personalized medicine with cell and gene therapy, where we harvest cells, we enhance them, and then we put them back in the original patient. And so there are innumerable challenges, both from a logistics point of view and a preservation point of view and an enhancement point of view. And so Chuck and Alison have really captured that. Bob has a lot of insight on this as well, especially from the gene therapy space. So Bob, if you could pick that up and continue to kind of frame this for our audience, please.
Yeah, I'd be happy to. And so you guys understand I'm working with Synthego, which is a genome engineering company that launched itself by building guide RNAs and progressively moved to build platforms that could augment the full path to clinic, GMP cell editing all the way through robotic or automated cell processing devices. So scaled up to us is not as complex because our piece of the pie is really biochemistry. It's chemical engineering rather than cell manufacturing. And we're feeding that component in as a GMP ribonucleoprotein complex or guide to be used to edit cell targets. But what we are doing is working with a lot of people that are looking at enabling that full chain. And what we see is a tremendous amount of pressure on reducing cost of goods to meet patient access requirements. People now exploring the procedure for delivery of a modified gene-modified cell target. And people are starting to look at procedures that can be simplified and used in outpatient setting to really routinize gene editing, particularly in blood. And so one of the things that we see as a consequence of that is that many of the patient access issues, take sickle cell disease as an example. You know, these are very expensive procedures to deliver a gene therapy product, which can actually be manufactured at very realistic costs if you can shift away from manufacturing using virus over to gene editing protocols. And so what we're seeing is that people are really focused on the holistic delivery path, that you've got to manage the cost of that whole delivery chain, or you're really not going to reach the goal of making these therapeutics accessible. And therefore, you're not really going to be driving your product or your confidence in your modality forward. So again, it's a really, really interesting space. The other complexity I see is that technology is moving so fast that it's outdating the commercial stream. And so many people are forced to launch into a long development process of pre-IND, IND enabling studies, and then going through a phase 1, phase 2 clinical trial scenario to get clinical proof of concept. During that 3 to 4 year period, you're seeing new technologies come up that really threaten competitively cost of goods or ways to deliver the modality. And so now the industry is caught on when they drop a current procedure and when they leapfrog or bring a new technology in. And so that's a wave of new companies that are embracing the new technologies that become, you know, a very positive threat towards, you know, the current commercial process. So it's really a very interesting space and a good one to be contributing to.
Before we move on, if you could talk a little bit more about that, because that sounds like a supply network challenge then. So there's innovations that are enhancing the process, and that's then just causing more disruption. And all of this at the same time with incredible capital outlay to get started. So how does that represent a challenge?
Yeah, actually, it's a chance for me to reflect this back in Q&A to Chuck and Alison. I mean, take CAR-T immunotherapies currently. You know, you have a very high cost of goods in manufacturing using lentivirus. You know, I know I'm pitching genome editing instead of virus, but we all understand, obviously, those reagents are much less expensive than the lentiviral space. So now you've got a competing drop going from $250K to $25K in cost of goods. There's huge pressure to deliver that into the commercial stream. But it's high risk for people that have developed infrastructure to practice viral-based gene delivery. How do you obviate your current product and bring a new product in? And even when you decide to do that, somebody else is behind your back jumping in. You're not going to be current even when you do it. So the cycle is moving, the development cycle, the regulatory cycle is too slow relative to the technology cycle.
Interesting. Let's get, now I want to move on a little bit to some solutions, just some of the things that Chuck and Alison and you have done in your experience to try to overcome some of these challenges. But I want to get back to that coordination issue, because what you just captured, Bob, was an industry coordination challenge, right? So let me shift over to you, Chuck, and then we'll talk a bit to you, Alison, and then you, Bob. But first, Chuck, so in light of the challenges we've just talked about, what are some of the things that KITE's doing, and what else have you seen that you think are some solutions to some of these challenges?
Yeah, it's a great question. Well, just building off of some of the commentary from Bob and from Joe, Joe mentioned about the access to viral vectors sometimes is a challenge at KITE. We are in the process of vertically integrating viral vector development, clinical and commercial manufacturing. So our clinical viral vector facility and pilot plant came online late last year, and our commercial viral vector facility will come online middle of next year. And that will give us the ability to produce our own retroviral vector and lentiviral vector for our development, research, and clinical programs, as well as commercial. So we're excited about that. Within the commercial manufacturing space, the vector, believe it or not, is not a big contributor to our costs. It's actually labor. You have to think about, we do, and Alison mentioned it earlier, the testing, if you will, the characterization of these products is challenging. So the QC testing is very laborious. There are certain assays that we use in the control system that are, specific for cell therapy, and they may be not as robust as some of the other tests that we've used for the traditional antibody products that many of us have grown up with. Manufacturing labor is also very intensive, which drives up the cost. So the majority of our cost is actually labor. It's not the vector or the raw materials. What KITE started to do is work with General Electric, which division within GE, to automate the manual process, thereby reducing the number of people we need per batch. And that technology will be inaugural in our manufacturing facility in Maryland, which will be licensed in the middle of next year. We anticipate being able to reduce our labor by about 50% in manufacturing. In addition, we have a lot of initiatives going on to bring a level of automation to QC that we don't currently have today. That's close collaboration between our global QC group as well as our analytical development group. And then we get into the next level of efficiency, which means the next generation of autologous cell therapy manufacturing process. So the automation I'm talking about right now automates the process that we currently have today. It doesn't necessarily improve it per se from a speed or simplicity perspective. But that's the next generation, which we have active work going on in process development. And now we're starting to have life cycle management conversations about bringing those technologies and innovation into the commercial products, in addition to obviously building those the next generation testing and manufacturing processes into the development programs and clinical programs. So that's kind of what we have going on right now. It's exciting. In addition, we're looking at taking the automation that we bring online in our Maryland facility and bringing it to the other facilities that we have in Amsterdam and in Southern California. So we believe collectively that will help improve the reliability, the efficiency, and reduce the overall cost of goods for autologous cell therapy.
Excellent. Thank you, Chuck. And we received a question that's related to what you've just said, and you've addressed it partially with what you said about automation, because certainly that improves quality, as you said. The question says, as chain of title is life or death in this space, what ways are companies looking to improve, control, monitor this need? Can you elaborate on that question a little bit for us?
Well, chain of custody and chain of identity is incredibly important. And I think what we rely on is the continual improvement, if you will, of our IT infrastructure to be able to do that. The IT systems that we use were innovated specifically for cell therapy. And we're in the process of launching our second generation of that chain of custody, chain of identity IT infrastructure. In addition, working closely with partners that we rely on from a transportation and logistics perspective to integrate our systems even more so than they currently are today. We've never had a mix-up. We don't intend to have another one, but we don't take it for granted. So we're constantly, Greg, it's a great question, whoever asked it, we're constantly looking at how do we make that better? Even though the current state seems to be just perfectly fine, we don't let up. We always want to push the envelope. We always want to get better, even when something seems to be working just fine.
Well, and I think as we talk about expansion, and the title of our session here is how to reach more people globally. I think that what you're talking about, Chuck, makes even more sense, because works great now, but as it gets spreads further and further, the systems will be tested more. So that all makes sense. Alison, if you could chime in here and talk a little bit about what Allogene's doing, and possibly if you have more to add on the question from the audience as well.
Yeah, I was just going to comment that chain of custody is a principle of GMP manufacturing that I think all of us, have been brought up with, and even simple breaches of chain of custody can be catastrophic, even with regular biologics, right? We've seen that in the news with Emergent recently. So I think that yes, circular supply chains where it starts with the patient and ends with the patient, there clearly is a higher bar there. We have a linear supply chain at Allogene. So we're dependent on those foundational chain of custody principles, in terms of GMP manufacturing. Beyond that, I wanted to pick up on what Bob said. So at Allogene, I think, and actually also what Chuck said. So, Chuck's already in the commercial space, and he described infrastructure and choices that KITE GILEAD have made in order to, globalize and scale out. The interesting thing, as Chuck referenced, is we are still in a very emergent technology space, and this is what Bob was talking about too. So, a lot of times I think we all get asked about automated workflows. Automated workflows are excellent if a platform is highly stable, robust, and it's the platform that you know for certain that you want to take forward and bank on for years to come. If you're working in a space where you feel there may be technology substitutions, then it may not be the right time to have integrated automated process flows. I think that in terms of solutions, I do see some vendors are providing quite good solutions in terms of independent unit automation capabilities, and we have certainly tried to take advantage of that at Allogene, but I'm not quite ready to just integrate our whole process flow, because I think that there's quite a lot of opportunity, and that's in the production space. The last thing that I'd like to say is, as Chuck mentioned, testing, I think in the traditional biologic space, the process is giant and costly. It takes a long time, and the testing really supports, the product that is generated from the process. It feels a little different to me in the cell therapy space. Of course, the process has its challenges, but the testing, the magnitude of testing, the duration of the types of tests, the heterogeneity of testing, ranging all the way from traditional testing, cellular, biological testing, and now very sophisticated molecular testing. So testing and thinking about duration of testing and automating testing flows is actually one thing that can make a very big difference in terms of speed and efficiency in cell therapy production flows.
Great. Thank you, Alison. That really helps. And thank you for the specific details. Bob, if you could pick this up, and I know you have a lot of insight on gene therapies, what's being developed to enhance gene therapies, and what's also being done to specifically address some of these chain of title or chain of ownership issues?
Yeah. I'll address chain of ownership a little bit less, just based on background. I think Alison raised a very important point about analytics that we should stay focused on. I think we underestimate the amount of retains and cost embedded in our cost of goods that go to lot release testing. And there's a move to use PAT or automated analytics during your bioprocess to eliminate the need for extensive lot release testing. In other words, are we now at a point where we can continuously track our cell editing and expansion process to a point that using machine learning and AI, we can prepare good statistical confidence ranges about lot release and not have to go through extensive time delays or cost delays in lot release? It's really a critical point. And I think particularly with respect to gene editing, we've got to understand how we can easily detect cited allelic drift in cell expansion, which is a background problem that we routinely have. And so if we probe our analytics too deeply, we're going to find changes that are not easily explainable, and it's going to slow the development process down. Let me go over to Chuck's point about automation on the process, and I think it's a tremendous move in the industry. I think people are moving away from patient-designated devices like the Multani device, and are breaking the unit operations apart and automating those so they can be used maximally for their purpose. And in doing that, and doing it in a clean room environment, you're starting to really be able to parallel process large numbers of autologous products in a very safe and reliable way. I like robots more than people in handling materials when it can get done. So I think the only other thing to add on is that there's a power building in linking process development across sites of work. In other words, using bioreactors or automated processes that communicate in the cloud to people using similar technology or similar devices. So you get real-time integration of data coming in. So you could have manufacturing sites in China, Japan, Europe, and the US, and you could have real-time integration of their bioprocess. And I think with AI and machine learning, we're really starting to understand the nuances at individual sites and can make big improvements there. So thanks.
So is what you're saying, Bob, I don't want to let you off this point here. So is what you're saying is then we network the learning, and then so from each of the sites, we're gaining insight and then sharing it?
Yeah, so take sickle cell disease as an example. You've got half a dozen people with viral delivery and correction of an HSC, half a dozen players doing gene editing approaches as well. 12 groups are trying to optimize cell collection, processing, expansion, delivery of that same potent HSC product. But nobody's sharing efficiencies or nuances in ways that AI and ML could easily call out some of the critical attributes that are responsible. So it's how do you parse out the common development, the virtues that you want to learn in these bioprocesses without compromising company or patient integrity. But there's a wealth of data we're accumulating that we're not mining. And now with AI and ML analysis of data, we can do that. Joe is in a wonderful position to be able to harmonize around that and understand the value of doing that. I trust.
Great. Thank you, Bob. That's a great cue for me, and that's what I was about to do. I was about to ask Joe, because now this is the second time a coordination issue has come up, and it's, I think, less in the kind of realm of an individual company to coordinate their own value chain, of course, but through an industry value chain. Joe, I know I'm putting you on the spot here, but what role could the City of Hope play and your organization play in these coordination issues? Bob's brought up the coordination issue around kind of a neural network of learning. And earlier, Chuck brought up the point, and Alison too, about this idea of coordinating technology advancement and also coordinating a wider supply network. So, yeah, I said that question over and over again to give you a little time to think through it, but I'd love to hear your thoughts.
Yeah, I appreciate that. I think that my take on this is highly informed by my position in the ecosystem. Let's put it that way. My job is to be a generalist. So we have to be able to take on things from all sorts of different types of products, different types of vectors, different types of cell therapies, modified or not. So we're more boutique. We don't have the benefit of being able to integrate things the way that the companies can, and I vastly envy that in a lot of ways, because we're still very much sort of, it's bespoke kind of cell therapy on a certain level. Our job is really to just prove that something is feasible and get you into your phase 1 process. It certainly is not likely to be the process you're going to take forward all the way to commercialization. There's likely to be changes. There's likely to be input, which is going to come as you do your clinical trials, give you better information on what to proceed. I mean, the best example of this certainly is like a potency assay. God knows nobody goes into these things with an appropriate potency assay, and everybody's terrified of it, since FDA is pushing down in these things farther and farther into earlier stage trials. But with more information from a variety of different sources, as you look at your CQAs and the way these things react in the body, you're going to have better input into what your potency assay should be.
So what you're talking about, Joe, then is more individual solutions for the multiple of companies that come to you, especially earlier stage companies, right?
Yeah.
So is your organization in a position to coordinate, though, or is that not your role?
I don't honestly think that that's the role we're best suited for. I think our goal is to get the training wheels off whatever process you've got, so that it's wobbling along and doing okay for what you needed to do. But ultimately, it's going to get more sophisticated. You'll be able to bring in more complicated techniques that truthfully, as you get beyond, say, phase 2, you probably want to go to a place which has more resources and more ability to do that. Our goal is really to get things into the clinic, and we leave anything commercial, for example, really to the big players right now.
No, that's fair. That makes sense. Go ahead, Alison.
I have a comment on that. I think that the idea that Bob raised is super compelling. I did have the opportunity when I was at Amgen to set up a global process performance sharing system where we had near real-time data being reported from all of our global manufacturing sites. And so the process engineers could overlay, current and historical data or multiple instances of current production runs. And the objective was to create, performance, process performance insights and learning from that. And that was really exciting. And for sure, what I take away from that is Bob's right. That's a great way to go. In order to maximize learning at this stage in the cell and gene therapy industry would require a huge amount of collaboration. I think that's the barrier. The technology is not the barrier. It's collaboration. And I think that what's quite shocking when one first enters the cell and gene therapy space is the N, goes way down, right? The number of patients treated, the number of production runs even is small compared to what I'm used to. And so to increase the N and to really maximize the learnings, requires some kind of cross-industry learning. There's quite a lot of discussion about that, but we're in a field that's highly capitalized and very competitive as well. So I think that continues to be quite a big barrier. The great thing that Chuck has, though, is, in this space, he has one of the highest ends of production runs, right? So I am sure that KITE is doing lots of run-to-run comparisons.
Yeah, thank you, Alison. That's excellent, and it's been a topic that we talk about at NIMBLE. Actually, is how can because NIMBLE is effectively an industry association, an industry consortium trying to solve industry-wide problems, and so it's. You can see some of the possible solutions. It could be driven by individual companies. It could be driven by industry consortiums. It could be driven by a government entity. And it's possible that different countries will choose different approaches with this whole coordination aspect. I'd like to shift a little bit, but stay on this topic of coordination because I think it's really important, Bob. Thank you for raising it, and Chuck and Joe and Allison for elaborating on it. This is LA best. This isn't USA best or even North America best. This is LA best. And what that means to me is that we're looking for regional solutions. And so I'd love for us to spend a little time talking about whether there are some regional solutions, especially in light of the fact that, at least in the literature, there's been a lot of talk about localization. Driven in part by scaling out in cell and gene therapy, but also 3D printing and kind of this mass local customization concept. So I'm wondering if there's something there, or if that's on the horizon for the cell and gene therapy, and why not LA? Because this is LA best. So Chuck, you offered to pick this one up first because when we talked earlier, this is a bit of maybe a provocative question. So if you could pick it up, I'd appreciate that a lot.
Oh, sure, Greg. Absolutely. The idea of having, just one manufacturing facility providing global supply of autologous cell therapy. As you start to stretch those supply chains between the apheresis treatment centers in one site, clearly, I think that's intuitively obvious that doesn't scale. I believe some of our competitors have tried that and realized that those are really put a lot of strain on supply chain. And we've seen other competitors go to more of a regional model that starts to look like ours. But even still, when you start to look at very broad and diverse geographical areas, like Asia is usually considered one territory or one region, but it's pretty big. If you try to draw a line from Melbourne to Tokyo, and we're starting to ask ourselves, what does the factory of the future look like? And what does our second generation autologous cell therapy process look like? And our analytical methods, does that enable a more dispersed manufacturing network where we could have less expensive facilities that are placed in countries able to turn around a CAR T in the same level of efficacy and efficiency and quality that we do currently in the U.S. and Europe, which benefits from, if you will, local geography. The other thing where we're seeing some differences in geography is certain countries are more open to CAR T clinical trials than others. So where else could we go in Europe and China, et cetera, even Japan, besides, in addition to the United States, and be able to then have a more diverse clinical profile, get more information, more data to better understand how our products perform in patients. So we're seeing a lot of opportunities, not only in the commercial space, but also in the clinical space. And it's getting us to kind of rethink, our clinical development strategies, as well as our commercial expansion strategies.
Great, thank you, Chuck. Joe, can you weigh in on this and this concept of localization? I mean, obviously, you're part of the ecosystem, as you raised earlier. I'd love to hear your thoughts.
Well, I'll take this on a slightly different tack, I think, which is that if we're looking at ways to keep the local LA biotech system healthy and functioning, continue to expand and to be the hub that it is, the thing that we can definitely work on is education of a workforce. We're all struggling to hire. There has to be a much more overt system in place, an outreach towards local colleges, local universities, training programs, because I guarantee you we can't feed the beast until we have well-trained people who are willing to do what it takes. And we are, at this point, we're all competing for each other's workers, and City Folks losing because we're not a company. Obviously, we can't kind of pay the kind of salaries that a company like KITE, for example, could pay. That's good luck to them. But the point is that we have to be able to support this whole industry locally with an appropriate workforce who can be depended upon.
Well, thank you for raising that. That's an excellent project proposal to Nimble for us to train people up within a particular geography. It's happening all across the East Coast. It's a new project we've funded in the San Francisco Bay Area. I think we should need a project like that here in Los Angeles. So thank you for that. Alison, your thoughts about the regional bit too, and then Bob, I'll come back to you. Sorry.
Well, so Allogene, headquarters is in the Bay Area, but we have many connections to the LA region. In terms of just production, we are running a production that is more centralized than it is dispersed. We are inventory generating, so that's a big advantage in the cell therapy space. So that means, so currently we have a plant in the Bay Area. Where we would put our next plant, I think, is going to be dependent on a lot of the things that Chuck was just describing. But I think in terms of just expansion of technology and talent, there's not much stopping us now following this interesting year and a half that we've had. And I think that what will happen in this space and what we're interested in is really following the technology. So, as LA continues to become a more and more vibrant technology hub, that's where we will establish satellites of business.
Great, thank you. Bob, I want to hear your thoughts on this for sure.
I don't think I have significant thoughts on this. It's a complicated question. So in the past, I've worked in Cambridge, Mass, and the environment there thrives because people communicate easily and casually. And so I think that's been a block for me in California in terms of particularly LA having, you can't drive an hour and a half to work every day. You have problems easily communicating. I think, Alison raised the fact that the last year and a half, we've learned to communicate over Zoom and maybe, eliminate the commute and the distance. So maybe there's really a power to come back and form networks that can thrive locally, but actually be dispersed. So I think it's a new model that we're coming up with how companies are going to form and accelerate. And you see people in their planning process really look to, as Joe's point is well taken, outsource manufacturing to a labor force and a region that can accommodate the kind of build that you have. You can't park that in the middle of San Francisco. So companies are building in a really different way. So this is a good topic to bring up for next year's LabVest panel. We should all write our predictions down about what the world will be in a year, and you can open them up and read them. So,
well, and our as all of us know, Mark and Judy have done a fantastic job organizing the conference with all of their colleagues. But one of the questions they frequently ask me is, what can we do to enhance the environment here in the LA area? And I think part of the answer is what we've been talking about. Hey, we only have a few minutes left, but I'd love for Chuck, if you could pick this up again and give us some of your closing thoughts on what's next. I think Bob started us, what's the world going to look like in a year? But just what are some of your thoughts and some of your kind of advice for where you think the industry needs to go for it to really optimize its success?
Oh, when we talk about the industry, that's a very broad term. There's a lot of different types of cell therapy modalities in various stages of development. We're talking right now about T cells, whether they be autologous or allogenic, but now there's also, iPSC, induced pluripotent stem cells. Which is beyond healthy donor. There's NK cells, whether they be healthy donor or iPSC. So I'm really excited to see in the future kind of where that science leads us. Does it take putting a CD19 CAR on a different cell other than a T cell? How does that change efficacy? And then what do you have to do to get something into with that type of a cell into an allogenic type of space? So I'm very excited moving forward to see what the science is going to uncover for us. I think Alison mentioned it earlier. The science is changing around us day to day. We are on the bleeding edge of innovation. And I'm really curious to see kind of where the science takes us as we move forward. At KITE, we're very focused on CD19 heme malignancies. I think a lot of other companies are too in the heme space. I also think solid tumor represents 80% of all cancers, and we really haven't been able to bring cell therapy to the solid tumors in the way that we have to the heme malignancies. So that's another area that I'm hopeful between TCR technology, neoantigen technology, that we can find a way to get to the solid tumor. We are tip of the iceberg with what we've been able to do so far. And the science is just so exciting. How do we pull that through, use other types of cells to go after different types of cancers, get away from the viral vector and use a different type of transduction medium to transduce the cells? I mean, it's just so much over the next 5 years, 3 to 5 years for us to understand and to see where it all takes us. It's going to be exciting.
Great, thank you, Chuck. Alison, if you could weigh in on a question that I get often, and that's about cost. And we've mentioned a number of things about the challenges of scaling and the challenges of integrating technologies for greater automation. Could you help us with this a bit? Where are we going to start meeting some, we'll call it exponential cost reductions? What do you see ahead?
Yeah, so I think Chuck earlier on talked about what a large commercial operation can really start to think about. I mean, a 50% reduction in labor is very significant in terms of translating through to cost of goods improvement. Just magnet, as in all manufacturing, utilization and scale, are often the biggest levers. So beyond that, in terms of order of magnitude reduction, yes, then I think we need to be looking towards some step changes in technology. So allogeneic technology should be able to deliver a significant step reduction. I already mentioned, Chuck has a process flow that goes from a patient back to a patient. So that's singular. I'm working on a process flow that can go from one donor to 100 doses. So that's different. And I think that there is much, much more potential for just scale in general, therefore access and availability of these very powerful medicines, to all who might need it. And then, of course, cost reductions.
Great, thank you, Alison. Joe, some final thoughts on the cost issue or on anything else?
Particularly, the goal for everybody for ultimate cost reduction is likely to be allogeneic. We all understand that. That's the best route forward in that case. Again, it depends on how much automation you can bring to bear in an early stage process, particularly for an academic group or a company that's just starting out. Are they willing to invest in a complex, highly automated process when their process itself is still somewhat in flux? So the best savings to me are going to come when allogeneic productions become routine, when allogeneic therapies are routine in that case. And but frankly, at just a little later stage in the development of the process than I do when we get to it.
Great. Well, thank you. I want to thank you, Chuck, Allison, Joe, Bob. Outstanding insight that you've given us. And I think it just really gets our audience up to speed. And also, we invite them to reach out to us if we can help as we extend this conversation. But thank you all for your excellent work. And thank you for being part of this panel.
And thank you, Gregory, for leading this very, very interesting panel. Thank you very much for everything you've done for us.